Hello, thank you for joining today's webinar, Digitalizing Sample Collection for Dynamic In Vivo Studies, presented by RockStep Solutions and Scientist.com. We have just a few reminders before we begin the presentation. This webinar is being recorded and will be made available to all registrants and attendees within 48 hours after the event. As we're all continuing to work from home due to the pandemic, we hope you will understand if we run into any technical issues or you hear background noise from the speakers. We appreciate your patience and your involvement. If you have any questions during the presentation, please ask them by using the Q&A function, which you should see at the bottom of your RingCentral window with the large letters Q&A. Our panelists will answer submitted questions at the end of the presentation. Before diving into our presentation, I'd like to provide an overview of the event today. As you can see, we'll start with an introduction of ourselves and our companies before we begin to discuss sample collection and digitalization and the relevant challenges and solutions. We'll finish up with the interactive Q&A session. I'd like to start by introducing myself and our other speaker. My name is Meaghan, and I'm the Scientist.com Director of In Vivo Services. Following my graduation with a bachelor's in agricultural sciences, I used my animal handling and welfare experience to work for Covance Laboratories in Madison, Wisconsin. During my 4 years working in the vivarium, I gained experience in many different specialty areas, including DMPK, ocular, infusion, DART, and surgical support. Once I completed my master's in biotechnology, I moved on to client services, but still spent time in the vivarium to keep up my skills. After 5.5 years at Covance, I was ready for a new challenge and moved to the Bay Area in 2017 to work with Scientific Software Solutions. I've been with Scientist.com since December of 2018 and enjoy working with clients and suppliers to advance animal welfare. Julie?
Hi everyone, I'm Julie Morrison and I'm president and co-founder of RockStep Solutions. And my background is actually in pathogenic and space microbial assay development at IDEX Laboratories and the Jet Propulsion Laboratory. And I've actually spent most of the last 4 years in vivariums and research labs across the country and even the world, researching and shadowing our customers and ensuring that the software we're building is actually what you guys need and it solves your problems.
Thanks, Julie. So I'd like to give you a little overview of Scientist.com. We're a B2E marketplace that was founded in 2007, and while we're headquarters in San Diego, we have offices and employees all over the world. We're currently supporting over 90 clients and over 3,400 suppliers utilizing our one pre-established contract model.
And RockStep Solutions is the maker of the software called Climb, which is for in vivo data and operations. Officially founded in 2016, we are the winners of 3 National Institutes of Health Innovation Awards for revolutionizing research workflows with future-proof technology.
And now on to our overview. Biospecimen or biological samples can include a wide range of things. The most obvious is blood that is centrifuged to separate plasma or serum. However, there are other types of collections that can occur, such as urine for clinical pathology, feces or bile for radio-labeled metabolism studies, or even milk for developmental and reproductive toxicity studies. In this instance, we're defining biospecimen or biological samples as any type of biological materials that are collected from animals for research purposes, and that includes cells, tissues, and excreta. Samples can be collected for many different purposes, and some are time dependent, such as blood drawn from animals on a study at certain intervals pre and post dose with test article. We call these time collections and the individual blocks time points, and some samples must be drawn during a certain window of time that's related to maybe sleeping or waking cycles, or even within a certain window of time after a technician enters a room. So there are also samples that are not time dependent as far as the minute or hour, but have to occur prior to another procedure being conducted. As you can imagine, it would no longer be possible to take a blood sample after an animal has undergone necropsy, for example. Finally, there are samples that are not strictly time dependent, but instead are collected in cycles, such as the blood sample that you might provide in an annual checkup. So challenges are huge in this area. What you see above the surface is a completed study report, but there's a lot that goes into that. One of the first things you learn in the vivarium is that you should always hope for the best, but prepare for the worst. Everything that a study team touches involves reacting to changes, and I've never seen a study go exactly as anticipated. You may have everything prepared, but a test article doesn't go into solution, so you can't dose animals on time. Or you might have a visitor observing the dose that requests a last-minute change. Animals can become sick or be replaced at the last moment. You might even be holding a dog for a dose and a tube slips and you get blasted in the face with group 4 test article. These are all actual things that happen to me in the lab. And luckily we're adaptable as humans. We can react quickly to these changes, but your paperwork doesn't react for you and your blood tubes don't relabel themselves. There's no automatic phone call that goes out to a study director to tell them that you're covered in all of the test material. So how do we manage this today and how did we manage it in the past? Well, usually responding to situations like these results in a crazy rush to get back on track. You're calling supervisors to get extra help. You're telling the study director there's a deviation from the protocol. And during all of this, there's somebody that's creating and printing new paperwork to record dose times and trying to figure out what all the subsequent collections need to be. At this time, they're also printing new blood and plasma or serum labels. They're trying to figure out who's available that can collect those samples late at night or on a weekend. And it's really not an easy thing to tackle, and it usually requires a team of seasoned technicians to sort out. Julie?
Thanks, Megan. So we talked about digitalizing sample collection, but what does that word even mean? So let's define digitalization by looking at this through the lens of an industry that's already gone through a full-scale, what we'd call digital transformation. So this is the airline and the travel industry. If you travel and you own a smartphone, you've likely been impacted by this. So in the 50s, airline tickets were handwritten. Perhaps a carbon copy existed somewhere, but it's unlikely that if you lost your ticket, you were going to get that back or recover your reservation. And at the very least, it would be incredibly difficult. This is what we call analog. Then came the printed tickets. You would need to arrive early. You had to go to the check-in counter, wait in line, have your security documents checked, get your ticket printed. You're still required to show this paper pass. This is what we call digital or digitized. Next came the kiosk. So we moved into more integrated databases that communicated with each other. You no longer had to stand in line to go to the specific check-in counter. You could check in right at this kiosk, scan your documents, your boarding pass would be printed. The machine would communicate all of this to all the other machines in the network, so the airline was aware of what you did at the kiosk without ever having to wait in line and talk to a person. This was called a digitalized system. Now, we check in online. We only show our security documents at the security checkpoints. Each airline has an app where a 2D barcode is rendered, replacing that paper ticket. Your phone communicates with the airline database, so you can check in without ever leaving your home. And not only that, but your phone uses artificial intelligence algorithms to find the best flight prices for you, the best rental cars, hotel discounts, and other items you need for your trip. You can even have a car waiting to pick you up by the time you left your airport. In fact, my colleague just did this and had his car ready, warm, and waiting for him in a cold Maine winter. So all of this happens through a digital transformation. This is an example of an industry all coming together. So now let's take that example and extract it into the life sciences industry. What does this mean for us? If you look at analog, this is your paper lab notebook. This is your sample collection paperwork. You may actually be in a database, but you still have to print everything out and then type it back in later. This is the analog. Digitized or digital is when you move that paperwork into spreadsheets or maybe an old siloed system. And then a digitalized system, what we're talking about today, leverages all of that digitized data and digital technologies such as API integrations to related databases or connectors to technologies like IoT sensors in your vivarium, machine learning or AI algorithms that tell you if they think a cage is being flooded and warn you preventatively. All of this improves the processes, drives efficiencies, and it does more with the existing data and enables new data. Now, when you put all this together, you have a digital transformation. And a digital transformation is part of an organizational ecosystem with really careful thought about how data flows between all of the different systems throughout the organization. The ultimate goal here is to drive business objectives with innovation and new digital technologies rather than just trying to improve all of these manual old technologies and ideally moving towards a more automated future.
Thanks, Julie. So now we're going to move on to talk about some challenges and solutions. First up is scheduling. So we want to discuss scheduling for technicians here. If you look at this chart, I've mapped out which species each technician has the ability to bleed or collect samples from, as well as which days and times they're available. In this scenario, they're rotating weekends and each one is taking a late night of sample collections. So how many studies do you think that these 4 could manage at once? And who would you choose to put on each study to manage it? Would they have to take all of the samples? Would they need to swap? And it gets really complicated, but now if you imagine there are 30 or more technicians that are collecting samples and each one is running 10 to 15 studies at a time, plus night and weekend collections, it's getting really, really hard to manage. So how would you make sure each study has all the sample collections accounted for and processed and stored in time? How would you even keep track of which studies need bloods and when they need them? The old school way of doing this is with cobbled together Access databases or giant Excel spreadsheets. And I used to actually have to do this. And neither of these options offer reliable tracking. There's no accountability and there's not very good security. It's also really time consuming, it's super confusing, and sometimes it's nearly impossible to do correctly. Julie?
So now let's look at the new school way of doing this, guaranteed to reduce Meaghan's baking bribery that she used to have to do to get people to take some of these time points. So the green color that you're looking at here represents a resource. So let's say this is Harry. If you look closely on this November 9th, you can see he has 2 studies scheduled to start at the same time. Now, Harry is scheduled to start these studies both at 9. We can actually look at a weekly view of this on the calendar in a database. And when we look at the weekly view, we're going to be able to see that we have some free time on a Tuesday. So we can actually take that dosing task. You can see both of those right on the left-hand side, side by side. We can take one of the dosing tasks and move it to a completely different day. And this is the anchor. So everything around that dose will actually move the entire study. Now, Harry can execute both studies on 2 different days, and we even scheduled it so his 24-hour time point from study 1 is not going to interfere with his dosing and time points on study 2.
So our next topic is scheduling animals for study activities, including these collections. And in this example of a small animal study, you can see that there are tox and there are TK animals in each group. And as you can imagine, all of the animals need to be scheduled for some sort of dosing. They would eventually have dose levels here, but only some of the animals need to have blood collected and processed. So the TK portion of the protocol tells you which animals need bloods at each time point. So if you look here, it tells the intervals and the animals per sex per group. So if you're only bleeding 3 per sex per group, there are a couple of ways to approach the dosing. You could dose the animals with blood collections up front and try to consolidate the bloods. But if you do this, you'll be bouncing between test material concentrations, and this is really a no-no. The result of this is that you're going to have gaps in your sample collection schedule no matter what. And figuring out when the samples need to be collected, in theory, based on how you plan to dose them, and what the gaps are going to be is actually a big headache. And you'll notice here that the controls don't even have the same time points as the other animals, which adds another layer of complexity.
Great, so here's an example of a digitalized study design plan. So I know there's a lot going on here because, as Meaghan noted, this is a pretty complicated study, but it was actually pretty simple to set up. So let's walk through it. On the left-hand side under that protocol column, you'll see that the experimental protocol is displayed. Now, this was designed as a reusable template, which is made up of tasks which are custom designed by your scientists and study directors. So that person is just going to take that and drag and drop it onto the screen, just like we saw with the scheduling. And then they're going to be able to drag the cohorts, which we'll see a few columns over, onto the protocol to enroll them on the study. Now, in this particular study, we had 2 different endpoints. So our TK animals were coming down after their last bleed while the tox animals were going out to full necropsy. So we've actually color-coded those to make it really clear which animals are being treated with which different endpoint. Now, the sample plan on the right-hand side under sample groups, this can be created on the fly, or it can actually be part of that same experimental protocol template if you're doing standard collections. And that just saves valuable time for your study director who's responsible for actually designing and scheduling all of this. So now let's move over to the schedule tab of this same view. So on this screen, you can still see the protocol and the task, but now we're more concerned with actually how to schedule this. So if you look at this dose, the way that we set this particular protocol up was that the dose is what we would call an anchor task. It just means that all of the other tasks are dependent upon when that dose is actually completed in that animal. So by scheduling the dose, and you can see here, we scheduled that at 7 a.m., all of the other tasks are going to automatically schedule around it. And that's the benefit of doing this in a database, because we've already told the system that 30 minutes before we dose, we need to take a microsample. 30 minutes after, we have a time point, et cetera, et cetera. So this schedules everything on the visual calendar that you were looking at earlier with that quick video snippet. So once this initial scheduling is complete, you can always come back and move this around, or you can go right back to that same calendar and drag and drop things to make them work for your organization. So speaking more about drag and drop, on this screen, what you're looking at on the left, so now my study director has already set everything up, everything's been scheduled by someone. Now I'm the technician. My role is to come in and actually execute this study. So on the left-hand side, we have all the study tasks queued up, ready to go for data entry. And we'll look at what those screens look like later. On the right, we have that visual calendar representation of the study. So if Meaghan ends up in a snowbank and can't get to the facility on time to start dosing, we can easily just drag that dose task to a new time slot or day, and the entire study would reschedule around it.
So next, we're going to talk about an example of a really complicated large animal study. If there are 30 or more animals in this study, then you can see that the sample collection is going to begin before the other animals have even completed dosing. And then the 1 hour samples are going to begin. So these animals are starting to overlap with processing and with the next blood starting up. And you need to note that there's also late night time points at 8 and 12 hours that will need to be scheduled for each animal. So now if you talk about these study examples, both of the ones that we gave you occurring on the same day, you can see that it's getting really complicated, but it's actually a common occurrence for them to overlap. So now you'll have to schedule all of these animals from different studies, different time points, and in different groups and subgroups. And it gets really difficult to keep straight manually, as well as, on paperwork. Usually your hand is going to cramp up at this point.
Thanks, Meaghan. So since this is a complicated study with a lot of different time points, let's look at different ways that you can view these task configurations. So here, when I promised you we'd look at what the data collection screen looks like, this is kind of your first peek at that. So here we're just looking at the dose task, and this is, depending on who is responsible for scheduling, this is really where you would come in to start seeing how things are lining up animal by animal. So we can determine, is it okay to dose our polyjuice potion as quickly as possible and just go animal to animal, or do we actually need to set specific increments between animals? And we'll look at that example in a moment. Next, let's add in our pre-dose samplings. So we know that we have to see all of our dose tasks. We have a bunch of early tasks that need to be considered for the light cycles. And by changing the dose time, the pre-dose due times are also going to change, because again, the system already knows that these have to take place 30 minutes ahead of time. So in this example, we're looking at these options so we can optimize our collections for light cycles.
Thanks, Julie. So the next challenge involves scheduling collections that actually are competing with each other. So this example is a dog ADME study with bile duct cannulation. And you can see a very basic timeline I made up for one dog here. So blood is collected pre-dose, 5 minutes, 10 minutes, 30 minutes, and an hour, and so on. But then bile is collected starting at 30 minutes, and then urine starts at an hour. So this is fine for one dog, but later time points are also going to involve feces and eventually cage wash and wipe. And if you take a look at the caging setup right here, you'll see that one dog is on top and one's on the bottom. So the tray underneath the cage goes to a tube in a funnel that brings the urine down to an Nalgene bottle that's nestled in a bed of dry ice. And the tube from the top is obviously going to block the bottom cage. And it must be carefully plugged up in order to open the bottom cage. And I'm guessing right now you're all thinking about how fun this looks, but I'd also like you to consider how many people are required to be in the room to complete these tasks for one dog, and that those people all have to be scheduled. So there's a doser, there's someone restraining a dog for the dose, there's someone collecting timed samples, and someone restraining the dogs for sample collection. And if you're lucky, you have someone recording the times for you. Oh, and by the way, don't forget to get the samples in the centrifuge within 30 minutes. So now if you add a second dog, because it was clearly too easy before, you'll see that the dose times for the second dog are going to start to overlap with the bloods for the first one. And you still have to get everyone in the centrifuge on time. So most studies have more than 1 or 2 dogs. You'll see here I've scheduled up for 4. And all of the samples are starting to overlap in this area. And you're also literally kind of doubling or tripling the amount of people in the room. And you have collections that are actually competing with each other because there are these overlapping bloods. And then there's even the urines and the bile and things like that. And you have to determine who's doing each and which one is most important. So when you're scheduling these out and actually starting to record these times, you can clearly see these overlaps and the issues are beginning to start here. This sheet is just for one type of sample collection. I set it up for blood, but it's already getting complicated. And these mistakes here, I didn't type in intentionally to prove a point. I actually made those just while I was working on this, but that's an issue we'll go through later.
Great. So in this study with our dogs, again, we're going to schedule around that dose. Now we have a couple of new tasks that we need to do before the study, before the dosing and after the dosing. We have some critical time points here. You can see that the blood, the bile, the urine are auto-scheduled around. So at the 1 hour blood collection, it's exactly 1 hour after the dose is scheduled to start. And these are because of the timings in the experimental protocol. Now, but in this case, Meaghan knows that the caging area is in tight quarters. In this example, what we looked at before was having every dose at the same time, and you just work through those as quickly as you can. But here, because it's such tight quarters, we know it's next to impossible to collect the samples from the cages and shimmy past the other technicians and the collection lines in less than 5 minutes per animal. So here, in order to accurately represent how many resources and time we need to conduct the study, we can actually take the dose time and we can set the increment that we want each of these doses to start at. So if you look at the second arrow on the left-hand side, you'll see that dose 1 is happening at 8 a.m., dose 2 is happening at 8:05, and so forth, etc. If you look above that, you can see that the system has automatically scheduled all of those early time points around those doses per animal. So we also see this type of scheduling for like really complicated takedowns when you only have a few resources available to help. And those doses must also be staggered to ensure there's enough time to necropsy each animal without any overlap.
So the final example we have of a challenge is scheduling for emergencies. It's definitely not a fun task, but it does happen. And one of my previous roles was to be on a response team for situations just like this. So you can see here an example of what must be done once an animal is determined to be moribund. There's clinical pathology, which involves tubes, labels, paperwork, and technicians, as well as necropsy, which needs a whole other suite of things. There's also a rather frantic call to a study director to inform them of what's going on, as well as the editing of all of the beautiful paperwork and labels and tubes that you might have prepared for that animal or group for the remainder of the study. And these all need to be either manually corrected and scheduled out or tossed out.
So in cases of emergency, using a digitalized system is going to make this so much less chaotic. On the left-hand side of the screen, we can actually run a quick search on the animal of interest, the animal that's moribund, very quickly pull up all of its necropsy tasks, or you could even quickly assign the new task if this was going on to an emergency protocol. And then on the right-hand side, you can actually select the sample records that are still not collected for the necropsy and quickly print those sample labels. So let's look at that in a little bit more detail. So here's an example. You're looking at that same sample screen here where your 3 samples are selected. You can actually send these directly to a printer. There's some examples of some sample labels there. Alternatively, you could export the sample information or use an API integration to send this all to a tube writer. So whichever way that your group does labeling is fine. And all of this happens in seconds to minutes. So now we have to deal with the quote unquote paperwork. So as Meaghan noted, this type of emergency could have ClinPath, necropsy, etc. We have a small example here, but the system can create all of these data entry screens in seconds, regardless of how many tasks you need to complete. And it really replaces the need to prepare and print any paperwork. So on the top left is an example of a real-time collection screen for the necropsy, where you can do your external examination and gross findings, and then actually mark off all the samples that you're collecting as well. on the bottom right, you have the timestamps of each task that's completed individually or in bulk. And because of the audit tracking and the chain of custody in the database, this is actually going to serve as your signature of completion. Now, on the right-hand side, the animal status and the exit reason are also updated in this screen. So when using a database like this value is going to auto-update in all the other areas of the application in real time so that all of the stakeholders like colony managers or study directors, vivarium technicians, et cetera, they all have these records updated without any extra effort on your part, and you don't have to field phone calls and emails from them wondering what happened to these animals.
Thanks, Julie. So the next main challenge we're going to talk about is dependencies or dependent collections. So in this example of a complicated infusion study, you can see there is a 60-minute infusion followed by a series of collections of different types. And I've just highlighted here a few of the most obvious dependencies. So you can see here the word pre-dose. That means ECGs obviously need to be collected prior to dosing. So do TK collections, some ClinPath, complement analysis, and urinary biomarkers. You can also see that there's a type of sample that involves fasting, as well as samples that require tube preparation before blood is added. So there's also a requirement for storage prior to shipment here. And it's not enough to just assume that these things are done. You need to note them somewhere to prove the dependencies were considered and that procedures were completed correctly and in the correct order. This is another example of a small animal study for DART. And so you can see here, there are additional dependencies. For example, there are common pre-study collections, which you'll notice are a theme, but there's also collections that are scheduled post-coitum. And you can see that there are necropsies scheduled to occur after the last blood sampling point. And this is really critical to note because it requires a high level of communication between different teams to ensure that they occur in the correct order. And as you can imagine, there is no going backwards on this one.
Thanks, Meaghan. So we've already looked at several examples of this kind of screen here where you have your to-do list for your study on the left-hand side, and I've just narrowed this down to the dose and all of the tasks that are due before it. And we're going to look specifically at one of those challenges Meaghan pointed out, the pre-study task. So here we need to do some baseline ClinPaths, we need to remove their food and do some blood collections. On the left, you can see all of that and ready to go into the data collection screen. But on the right, you can see it actually on the calendar. So let's say that this dose is scheduled for Saturday, and we'll keep with our Harry Potter theme. This is the night of the Hogwarts Winter Ball, so nobody wants to be in dosing that day. But we can't just move the dose because the study obviously has these critical baseline tasks before we ever start dosing. So what we'll do is actually just take that dose task, pick it up, and drag it 2 days over. And this requires that all of the pre-dosing tasks move up as well. So with this type of drag and drop scheduling, a digitalized system will do this for you.
Thanks, Julie. So the next challenge we're going to discuss is preparation and processing of those samples that you're collecting. So this is an example of a pre-printed collection sheet that has some fairly common information outlined on it. You can see there's space for study number, sample type, volume, and site, as well as entrance of the actual dosing time periods. So I've added the study number and the sample type and all the current information here. And the intended site of collection is included just to help technicians work through it. So with this paperwork, you need to be preparing it for all of the animals at each time point. And for basic large animal PK studies, it's not overly complicated because each animal gets a sample collected at each time point. But if you have small animals with subgroups, this can get messy really, really quickly. And here's a more complicated example. So continuing on with the infusion dose from before, this is an actual real study outline. And it involves 36 animals. So you can see you can fit 3 different time points on this paperwork, and you can actually fit about 20 animals running off the bottom of the screen here. So if you think there are 11 collections, when you count up these time points on the 2 intervals, that's 4 pages of time points with 2 pages of animals and 2 intervals, or 16 pieces of paper that need to be prepared and filled out individually. So if you think about equipment or apparatus, there's also preparation that needs to occur. And in this continuing example, we're talking about a blood tube as well as a tube for plasma or serum. And as you can see, there's a fair amount of information that you need to fit on the label for these tubes. And it includes study number, identifier, sample type and interval, the time point, and the actual contents of the tube. So it doesn't really look that bad until you think about how many you actually have to prepare. So 36 animals at 2 intervals with 11 time points and 2 tubes each is nearly 1,600 labels that you're responsible for creating and then placing on the tubes. And completing this correctly in an Excel sheet and then exporting it to Word to print labels is not really ideal. Your technicians prepping and your QC people are not going to thank you for doing this. So finally, in processing, it's its own timing challenge. So serum samples that are allowed to sit less than 30 minutes are likely to retain some cellular elements and other contaminants that are going to impact your future analysis. Samples that sit over 60 minutes are likely to experience lysis of the cells in the clot, and that's going to release cellular components that you shouldn't be able to find in your serum samples. So for tubes with anticoagulant being processed in plasma, that either needs to happen immediately or the samples need to be chilled and then spun within 4 hours. And these requirements are really critical to maintaining your sample and data integrity. And again, these steps need to be scheduled properly and then documented. So the old school way I showed you previously, and it's not hard to guess, it's by doing this by hand on paper.
So let's talk a little bit about this dreaded paperwork. As we saw earlier, paperwork is going to be replaced with these collection screens auto-created by the system. Now on this particular screen, you're actually looking at your pre-dose collections, your doses, and your post-dose collections. And by arranging them all on the screen at one time, you can actually ensure that your overlapping and competing time points are not going to be either scheduled at the same time, or if they are competing, that you're very aware you need to have more resources in. So now, in addition to lining up those time points appropriately, making sure that we know what order to accomplish everything in, the technicians also have detailed task instructions. So on the left hand, where it says like blood collection tube, you can get these detailed task instructions like what tube to put it in, or what anticoagulant, or for dosing, what the test article is, what the dose concentration is. And then on the right-hand side, they also have very detailed sampling instructions right in that screen. So if something is critical to go on wet ice immediately, it's specified right here on the screen for the technicians. All right, so another part of the preparation is around scheduling. So how many of you have created complicated spreadsheets to figure out how to schedule a study to avoid these overlapping collections? I've seen some pretty gnarly ones over the last few years. Well, as we saw earlier, the digitalized system can help your scheduler ensure that these competing time points are accounted for. So in addition to collecting your data here, the scheduler can come in and see how these are lining up and account for any of these staggered time points to make sure you have staff on hand. So all of this can take place right on the collection screen, and all of the time points can be put in one place, just like we saw earlier. Here we have all of our pre-tasks. We'll line those up with our dose task. And do you remember when we had the scheduling protocols in the study screen? Well, those experimental protocols are created by the study directors, and they are the group that is ultimately responsible for ensuring that these time points are collected appropriately. So this is an example of a screen looking at all of your doses together that can really help them come in and see how everything lines up. All right, so we've been looking at how the study director brings those study design or experimental templates in. We've been looking at how the scheduler and the technician goes into that collection screen to either refine the schedule for overlapping time points, or actually could do the collections and do your signature there. But how is that all actually created? So as Meaghan said, there's a lot of times where you may need to bring in centrifuging and things that are very, very time sensitive to make sure that your blood samples and your other samples are handled properly. So what you're looking at here is actually kind of the bones of that. This is where your study directors are going to create their experimental templates. These are created once, they're used over and over again. They can be made as vague or as granular as needed. So they can specify when to sample, when to centrifuge, when to harvest, to really ensure that all the specimens are viable when processed. And then this experimental protocol just gets added to the study. Once it's scheduled, it's visible on that calendar we looked at. And then on the collection screens that we just viewed, where all of these special processing instructions will be available. So this kind of completes the full circle.
So our next and our final challenge is the data input. While the input is our focus, we also need to consider error as well. And obviously, I'm not a robot, so I made some mistakes when I was originally typing this out, as you can see here. I did the same thing when I was handwriting data. We called the notes we made for these deviations e-notes, which is short for electronic notes. And I was very, very good at writing e-notes. So what we would do is come back in and look at the footnotes that we have here. And let's add another level of complexity to that. It's a rather simple study schedule, but there are some issues. I recorded all the times correctly when I was actually writing them, but there's another problem. So as you can see in the circles, we were late with sample collection. If you look at animal 2, we started being late at the 5-minute time point. So we were able to get back on track, but then at 10 minutes, the animal maybe had some emesis. And this doesn't necessarily mean we need to get a study director, as the animal's probably low dose, but maybe the dog has a sensitive tummy. And the time we spend dealing with this delayed the next collection as well. But then when you get to the half hour, we have to collect urine. And if there's been emesis, the urine's probably contaminated, and now we have a huge issue that's also delaying the next collections. So this threw off basically the entire schedule. And as a technician, you can write this down and make it look nice where nobody would notice, but you have to tell someone that a deviation has occurred. So I've notated the raw data now with my footnotes and gone and flagged it to direct them to my study note or my electronic record of what happened. And the system I used was exactly this. I wrote times on paper, then afterwards made a footnote. I had to call the study director, and then I went to a room somewhere with a computer and created an electronic note about whatever happened that caused us to be late. But then what? As a sample collection technician, you can pretty much just walk away. As a study coordinator, a director, or maybe even a report writer, you need this to be called out for you in a clear way. And as I like to tell people, you don't know what you don't know. So you need all of these things to be flagged. And then there's a clinical example here of data input, but it's basically the next step in the process of data entry for these studies. So after samples are processed and the testing is complete, somebody has to enter all of these values back into a spreadsheet. And you can see there's an identifier here for a patient, but in our case, this would be an animal. There's the study day, there's the scheduled time, the actual time, and then the concentration. And this is another opportunity for human error when you're entering this information.
So we've designed the study, we've scheduled everything in just the right way, we've staggered all of our doses here by 5 minutes, we have enough staff on hand to accommodate for overlapping time points. Each technician can have their own version of the screen on their own device. They're going to autocomplete and sign off on their task. Everything's going to be perfect, right? Well, there's a reason that we call this talk Digitalizing Sample Collection for Dynamic In Vivo Studies, because we all know things change quickly when working with animals. So in this screen, you can see that the top tasks that are now grayed out have been completed, but now at our half an hour time point, we're starting to get a little behind. So the system is going to point that out for us that animal 2101 is falling behind. Now, when your time points are collected late, technicians can actually write permanent notes associated with the task. There's no need to cross things out, initial them on the written paperwork, go to a back room, write up a report after the fact. This can happen on the fly. And because each person has a unique login, this record is audited with chain of custody to the person that was logged in. So there's no more handwritten initials. Now, the database also records a count with each note, and the notes typically signify that there is something that needs to be reviewed. So the reviewer can actually come in situ in real time and review these notes. Now, the reviewers and the study directors can also look at the results in QC in real time. So let's orient ourselves here. On the left-hand side is that same data collection screen we've been seeing. So technicians are going here, they're clicking off, they've just done the dose, now they're gonna go to the next bleed. But on the right-hand side, the reviewer might actually have up this screen, which is actually showing a running count of how the study is going. So you can see the top screen, the top few tasks are complete, as we saw on the other screen, but we're also seeing that there's some real deviations happening here. Well, the deviation between the due time for that animal and the complete time is automatically calculated here. And it also can be QC'd from any device, because this will pop up wherever someone is. So they don't, you don't have to be in the vivarium, you don't have to be in the necropsy room or wherever you are doing this work. And imagine if the reviewer could be at their desk or even at a different facility and intercept a lagging study and send additional help immediately. So the other thing we want to look at is a similar screen. Now on the right-hand side, we've set it up. We still have our running log of how the study's going. So you can see we have a lot more tasks that are completed here. On the left-hand side, the study director might want to look at the note count by those deviations, because remember, notes are generally means that something has gone wrong. They can sort on that and then go in and actually view those, look at this, QC it in real time, and export these to create their study reports.
So you can complete all of these difficult manual tasks that I had to do and tackle your challenges using the digital solutions between Scientist.com and RockStep Solutions. All the way from planning to purchase, Scientist allows you to have visibility, traceability, and control over your studies. And then when you use Climb by RockStep, you can conduct the studies with a high accuracy and create schedules with ease. You can actually purchase Climb through Scientist.com for a true end-to-end solution. And digitalization for the in vivo study lifecycle is really at your fingertips now. So thank you so much for joining us today. We are now going to switch over to the last portion of our event, which is the Q&A session. Please remember that you can submit your questions in the Q&A box. Julie?
Yes, thank you very much. So we did have a question come in earlier that we answered around, would these slides be made available after the fact? And yes, the recording will be made available within 48 hours and emailed. Just looking at our questions here. All right, so how do you determine recommended spacing between collections? I think that's a Meaghan question.
Definitely, thanks, Julie. So recommended spacing for collections has to do with a couple of different things. And one of those things is your technicians and their comfort level and how many hands you have on deck. So if you only have 2 people to catch a room of primates, you're probably going to need something more like 3 minutes in between. But if you maybe have 4 people, you might be able to dose every minute. So technician comfort and staffing level is really important. The other thing that's really critical is considering those additional samples. If you're just collecting blood, you can probably go through really quick, but as you can see, once you're starting to add sample collection types and all of those samples need to be collected in a certain order within the same minute, you really start to have to scramble. And so you really don't want to be stressing your technicians by scheduling those one after another, because it's definitely not going to happen and you're going to start to deviate really, really quickly.
Great, thanks, Meaghan. And just to remind everyone, there is a Q&A button. If you hover over the bottom of your Ring Central screen, you should have a menu of icons. There's a big Q&A there. Feel free to submit your questions. We have a couple of those that have come in. Our next one is, do you ever have issues with animal identification when you use paper methods? Do you want to start that one, Meaghan?
Sure, I think we can tackle this one together. So yeah, when you're working on paper, it's really easy not only to write down dose times or collection times in the wrong space on those huge grids, but it's also really easy to sometimes misidentify animals. So one of the things that you can implement in the lab is maybe chips or identification. And Julie, I know that you guys have a cool solution for that.
Yeah, actually, so kind of your first line of defense is really in that data collection screen. So the housing ID, some of the animal information will be displayed there as well. So you at least can narrow down to where the animal is located. But then you also have the ability to scan the animal. So if you're using, if it's mice and you're using like those rapid ID 2D ear tags that are color coded, it will display the color coding as well as the rapid ID information there, so you know which mouse you're picking up based on the color. There's also a place, we didn't look at this in the data collection screen, but you can scan that in. So if you have an RFID chip or a rapid tag, you can scan that chip into the database, and it will give you a check mark if it matches the animal that's scheduled, and it'll give you kind of like a big caution X if it is not matching the animal that was scheduled for that task. So just another line of defense to make sure you have the right animal.
Yeah, and that's really good to hear, because as a technician, I definitely fasted the wrong animal more than once, and delayed surgeries and other things. So it's really critical that you're identifying correctly and using that kind of digital backup instead of just taking a glance at a tattoo or an ear tag.
Yeah, for sure. All right. Next one, do you recommend printing sample labels before you start collections or on the fly to ensure the labels match the samples? And then there's a follow-on question about printers. So I think we can tackle the first one. So maybe we might have different answers. I can take it first, Meaghan. If you want to jump in, feel free.
Sure.
So I think it depends a little bit on your SOP and your protocols. I generally recommend that people print as close to the time of necropsy or collection as possible. And that way you're not creating sample records with a chain of custody in the database that have to be deleted if something happens. You don't have extra tubes or labels. You're not doing all that work. With that said, I have seen organizations, particularly if they're closer to GLP, maybe not quite GLP yet, but preclinical, and there is no matter what, if the animal is moribund, they are still collecting these same samples. It does not matter. There is no different emergency necropsy. I have actually seen them print everything on day one, and then it's all prepped and ready to go. So when you do have that moribund animal, you rush it and everything's already there. If your animals are going on towards a normal necropsy, but then you have a separate emergency necropsy if something were to happen, then you could reconsider printing them closer to the time of collection. And I see that happen anywhere from, one group does it every Monday for everything they need for the week. Others have a policy to do it no more than 24 or 48 hours ahead of time.
Yeah, I usually would make sure everything I had was set up at least 24 hours ahead of time. The exception to that is if you saw those time points on the paperwork. If time points were something crazy, like one was day 30 and one was day 60 and day 90, I would have that all set up just because it was on the same sheet of paper. I think it also depends on what kind of study you're doing. If you're expecting there to be a moribund group of animals, or you're doing something highly experimental, it definitely doesn't make sense to set that up completely ahead of time. But it is important to consider your internal SOPs, and if you need to have things QC'd by another member of your team. Our policy was always to have somebody that was specifically trained in reviewing paperwork and labels to look that over, and we tried to give them as much time as possible.
Cool, thank you, Meaghan. And then the follow-on question, what label printers do you recommend? So I can give some examples of what we've seen with folks using Climb, and then Meaghan, feel free to jump in as well. We see dymos a lot, both the regular and the twin turbos. If you are using something like a rapid tag and you want to see the color of that tag, then you do need to go to a printer, something like a Zebra, which allows you to print in color. I would say those are probably the two most popular I see.
Yeah, since I was using the old school method, we had some very old, sad laser printers and label paper. So I definitely don't have a recommendation other than not that, please.
Cool. All right, we have another question that's come in. Does the system accept external reference lab data or export collection data to an electronic data capture system? So it looks like basically, can you import data and then export? So the answer to that is yes. There's a couple of different options there. If we're talking about Climb can do a couple of things. So what we would call the old school way, which is actually still newer school than what Meaghan was doing, would be to set up, we didn't really go into how the task on those experimental protocols were set up, but if you remember right, like you could collect data like the body weight, for example. So all of those are going to be custom templates. So you can actually set that to match the CSV or Excel file coming off of your assay, or if it's for the reference lab, whatever their paperwork looks like, so that you can drag and drop that file in. The system will parse it, and then all of that will be in the system. It's a pretty slick way of just drag and drop and getting all the data in. The newer school way, which we're really implementing now and working with some vendors on integrations, would be using the API. So truly becoming part of that network where your ordering comes in from scientists, and then you run all of your end-to-end birth to freezer, I like to say, things in climb, and then from there you send things out to either your CROs for assay analysis or your reference labs, and then you bring them back. And then that probably needs to go to somewhere else, into an electronic lab notebook, or go further upstream.
Yeah, Julie, there's a really interesting question for you. Can you add your own measurements other than the normal ones like body weight or blood? For example, something like light exposure or music in decibels or anything?
Yeah, for sure. So similar to what I just mentioned with the creating your data outputs to be whatever you want them to be. So I don't know if it was an ELISA coming back, you might have a column for the well position and then each of their different concentrations. You can also create really anything you want. So it can be body weight. It's a pretty flexible platform, so it can work across different therapeutic areas, different tasks. So the short answer is yes.
I think this is also a really good way to keep track of enrichment and animal welfare activities as well. I know that, we all have SOPs about how often to give, new huts to our rodents or fruit to our primates. And I think it's important to note that you can enter these things in climb as well, as part of, just your regular protocols to kind of make sure everything is flowing along. And Julie, I assume that means you could, enter type of fruit given and make sure you're varying things.
Oh yes, absolutely. That's such a great question and a good point. We've definitely seen this with some of our, we've one pharmaceutical company in particular that has mentioned they're having some of their animals are housed at a vendor, and they have a really difficult time writing their animal welfare reports because they're not sure what kinds of enrichment and things are being given. So that's actually a use case that we're working on right now. And yeah, you can definitely pull a report up and go see, what you need to vary, how often. We had a actual, at one of our groups, they had what they thought was going to be like an outbreak. Really, the rats had all these pressure pad, or like pressure sores on their paw pads. And it turned out they had their little red plastic huts, and it was like monkey see, monkey do. They just started bouncing on it and it was fun. And then the other rats saw and they started bouncing. And I guess they just did it so much until they gave themselves these sores. And so in the database, you can, mark that off and make sure that that is noted to never use the red pressure huts in that rat room again.
Well, that's a crazy example. I've seen monkey see, monkey do actually happen in the lab, but I didn't know rats did the same thing.
I did not either. All right. Let's see if we have any other questions that have come in. I'm just reading through these.
Julie, I can help you out. I see another one.
Okay, cool.
Do you experience all study directors and managers interacting with the system, or do you end up targeting a few ones with the right mindset that become responsible for all of the studies?
Yeah, I think in general, it's better when everyone is using the system because each person has their login, each person has their own audit, and it becomes generally more efficient for, obviously we're trying to improve processes, improve efficiencies, and it really helps when everyone is on target. Now, the caveat I would say to that is if you're piloting a software, you do want to take, a couple of people that have the right mindset, that are really interested in working on this and making sure that this change goes forward, to become your primary stakeholders and your, what we'd call like super users that are going to help drive this throughout and really kind of become your go-to person in-house.
Yeah, and I think we do that same thing as well. People are sometimes really resistant to change. They've been doing the same thing for a long time. And so when you're doing a pilot, and you find someone that's really receptive to it, that can be kind of your champion moving forward. They can help evangelize within their organization. And usually it doesn't take too long for things like this to start catching on. I know if Julie had come into the lab where I wasn't showing me this, I would have been shouting to the rooftops for everybody to hear that I wanted to use this instead of the paper. So if you can find a few key people, and help move things forward, whether it's for digital purchasing systems or digital tracking or logistics or whatever it is, I think that's always a good thing.
Definitely.
Yeah. So Julie, I'm very sad that I didn't meet you and hear about this 4 years ago. Excellent. Well, I think that's actually all of the questions. So we're going to go ahead and wrap up. Thanks so much, Julie, for presenting with me. And thank you to the audience for attending as well. If you have any additional questions or you'd like to get in touch with us at scientist.com or at RockStep Solutions, our information is available on this slide. And as a reminder, we did record this webinar and it will be available for later listening. So thank you again for joining us, and we hope you found the presentation useful. Thank you.
Thank you, everyone.